So hello everyone and welcome back to NEETOPEDIA, that is NEET ka encyclopedia. And today we are back with our biology audiobooks after a long time. We will be continuing with plant physiology first chapter, that is transporting plants. Also do not forget to check out the other NCERT audiobook playlist of physics and chemistry on our channel. Now without wasting our time, let's begin. Chapter 11, Transporting Plants. Have you ever wondered how water reaches the top of tall trees, or for that matter, how and why substances move from one cell to the other? Whether all substances move in a similar way in the same direction, and whether metabolic energy is required for moving substances. Plants need to move molecules over very long distances, much more than animals do. They also do not have a circulatory system in place. Water taken up by the roots has to reach all plants of the plant, up to very tip of the growing stem. The photosynthetates, or the food synthesized by the leaves, have to be moved to all parts, including the root tips embedded deep inside the soil. Movement across short distances, say within a cell, across the membranes, and from cell to cell within the tissue has to take place. To understand some of the transport processes that takes place in a plant, one would have to recollect one's basic knowledge about the structure of the cell and the anatomy of the plant body. We also need to revisit our understanding of diffusion besides gaining some knowledge about chemical potential and ions. When we talk about movement of substances, we first need to define what kind of movement are we talking about, and also what substances we are looking at. In a flowering plant, the substances that would need to be transported are water, mineral nutrients, organic nutrients, and plant growth regulators. Over small distances, substances move by diffusion and by cytoplasmic streaming, supplemented by active transport. Transport over longer distances proceeds through vascular system, the xylem and the phloem, and is called translocation. An important aspects that need to be considered is the direction of transport. In rooted plants, transport in xylem is essentially unidirectional from roots to the stems. Organic and mineral nutrients, however, undergo multidirectional transport. Organic compounds synthesized in photosynthetic leaves are exported to all other parts of the plant, including storage organs. From the storage organs, they are later re-exported. The mineral nutrients are taken up by the roots and transported upwards to the stem, leaves, and the growing regions. When any plant part undergoes senescence, nutrients may be withdrawn from such regions and moved to the growing parts. Hormones or plant growth regulators and other chemical signals are also to be transported, though in very small amounts, sometimes in a strictly polarized or unidirectional manner from where they are synthesized to other parts. Hence, in a flowering plant, there is a complex traffic of compounds moving in different directions, each organ receiving some substances and giving out some others. 11.1 Means of transport. 11.1.1 Diffusion. Movement by diffusion is passive and may be from one part of the cell to the other, or from cell to cell, or over short distances, say from intercellular spaces to the leaf to the outside. No energy expenditure takes place. In diffusion, molecules move in a random fashion, the net result being substances moving from region of the higher concentration to the region of the lower concentration. Diffusion is a slow process and is not dependent on a living system. Diffusion is obvious in gases and liquids, but diffusion in solid is more likely than of solid. Diffusion is very important to plants since it is the only means for gaseous exchange within the plant body. Diffusion rates are affected by concentration gradient, the permeability of the membrane separating them, the temperature and pressure. 11.1.2 Facilitated Diffusion. As pointed out earlier, a gradient must already be present for diffusion to occur. The diffusion rate depends on the size of the substances. Obviously, smaller substances diffuse faster. The diffusion of any substance across the membrane also depends on its solubility in lipids, the major constituent of the membrane. Substances soluble in lipid diffuse through the membrane faster. Substances that have hydrophilic moiety find it difficult to pass through the membrane. Their movement has to be facilitated. Membrane proteins provide sites at which such molecules cross the membrane. They do not set up a concentration gradient. A concentration gradient must already be present for molecules to diffuse even if facilitated by the proteins. This process is called facilitated diffusion. In facilitated diffusion, special proteins help move substances across the membrane without expenditure of ATP energy. Facilitated diffusion cannot cause net transport of molecules from a low to high concentration. This would require input of energy. Transport rate reaches a maximum when all of the protein transporters are being used. Facilitated diffusion is very specific. It allows cell to select substances for uptake. It is sensitive to inhibitors which reacts with side protein chains. The proteins form channels in the membranes for molecules to pass through. Some channels are always open, others can be controlled. Some are large, allowing variety of molecules to cross. The porins are the proteins that form large pores in the outer membrane of the plastids, mitochondria and some bacteria, allowing molecules up to the size of small proteins to pass through. Figure 11.1 shows an extracellular molecule bound to a transport protein. The transport protein that rotates and releases the molecule inside the cell. Example, water channels made up of 8 different types of aquaporins. 11.1.2.1 Passive symports and antiports. Some carrier or transport proteins allow diffusion only if two types of molecule move together. In a symport, both molecules cross the membrane in the same direction. In an antiport, they move in an opposite direction. When a molecule moves across a membrane independent of the other molecules, that process is called uniport. 11.1.3 Active transport. Active transport uses energy to transport and pump molecules against a concentration gradient. Active transport is carried out by specific membrane proteins. Hence, different proteins in membrane play a major role in both active as well as passive transport. Pumps are proteins that use energy to carry substances across the cell membrane. These pumps can transport substances from a low concentration to a high concentration, that is, uphill transport. Transport rate reaches a maximum when all the protein transporters are being used or are saturated. Like enzymes, the carrier protein is very specific in what it carries across the membrane. These proteins are sensitive to inhibitors that react with protein side chains. 11.1.4 Comparison of different transport processes. Table 11.1 gives a comparison of the different transport mechanisms. Proteins in the membrane are responsible for facilitated diffusion and active transport, hence show common characteristics of being highly selective. They are liable to saturate, respond to inhibitors, and are under hormonal regulation. But diffusion, whether facilitated or not, takes place only along a concentration gradient and does not use energy. 11.2 Plant-water relations. Water is essential for all physiological activities of the plant and plays a very important role in all living organisms. It provides the medium in which most substances are dissolved. The protoplasm of the cell is nothing but water, in which different molecules are dissolved and suspended. A watermelon has over 92% water. Most of the herbaceous plants have only about 10 to 15% of its fresh weight as dry matter. Of course, distribution of water within the plant varies. Woody plants have relatively little water, while soft part contains mostly water. A seed may appear dry, but still it has water. Otherwise, it would not be alive and respiring. Terrestrial plants take up a huge amount of water daily, but most of it is lost to the air through evaporation from the leaves, that is, transpiration. A mature corn plant absorbs almost 3 liters of water in a day, while a mustard plant absorbs water equal to its own weight in just about 5 hours. Because of its high demand for water, it is not surprising that water is often the limiting factor for the plant growth and productivity in both agricultural and natural environments. 11.2.1 Water potential. To comprehend plant-water relations, an understanding of certain standard terms is necessary. Water potential is a concept fundamental to the understanding of water movement. Solute potential and pressure potential are two main components that determine water potential. Water molecules possess kinetic energy. In liquid and gaseous form, they are in random motion that is both rapid and constant. The greater the concentration of water in a system, greater is its kinetic energy or water potential. Hence, it is obvious that pure water will have greatest water potential. If two systems containing water are in contact, random movement of water molecules will result in net movement of water molecules from the system with higher energy to the one with lower energy. Hence, water will move from the system containing water at higher water potential to the one having low water potential. This process of movement of substances down a gradient of free energy is called diffusion. Water potential is denoted by the Greek symbol psi and is expressed in pressure units such as Pascal. By convention, the water potential of pure water at standard temperature, which is not under any pressure, is taken to be 0. If some solute is dissolved in pure water, the solution has fewer free water molecules and the concentration of water decreases, reducing its water potential. Hence, all solutions have a lower water potential than pure water. The magnitude of this lowering due to dissolution of a solute is called solute potential or psi s. Psi s is always negative. The more solute molecules, the lower is the psi s. For a solution at atmospheric pressure, psi w is equal to psi s. If a pressure greater than atmospheric pressure is applied to pure water or a solution, its water potential increases. It is equivalent to pumping water from one place to another. Can you think of any system in a body where pressure is built up? Pressure can build up in a plant system where water enters a plant cell due to diffusion, causing a pressure build-up against the cell. This makes the cell turgid. This increases the pressure potential. Pressure potential is usually positive, though in plants, negative potential or tension in water column in the xylem plays a major role in water transport up a stem. Pressure potential is denoted as psi p. Water potential of a cell is affected both by solute and pressure potential. The relationship between them is psi w is equal to psi s plus psi p. 11.2.2 Osmosis. The plant cell is surrounded by a cell membrane and a cell wall. The cell wall is freely permeable to water and substances in solution, hence is not a barrier to movement. In plant, the cell usually contains large central vacuole whose contents, the vacuolar sap, contribute to the solute potential of the cell. In plant cells, the cell membrane and membrane of the vacuole, the tonoplast, together are important determinants of the movement of molecules in or out of the cell. Osmosis is the term used to refer specifically to the diffusion of water across a differentially or selectively permeable membrane. Osmosis occurs spontaneously in response to a driving force. The net direction and rate of osmosis depends on both the pressure gradient and the concentration gradient. Water will move from its higher region of chemical potential to its region of lower chemical potential until equilibrium is reached. At equilibrium, the two chambers should have nearly the same water potential. You may have made a potato osmometer in your earlier classes in school. If the potato tuber is placed in water, water enters the cavity of the potato tuber containing concentrated solution of sugar due to osmosis. Study figure 11.3 in which the two chambers A and B containing solutions are separated by a semi-permeable membrane. Number A. Solution of which chamber has a lower water potential? Obviously, it's chamber B. Solution of which chamber has a lower solute potential? That's again chamber B. C. In which direction osmosis will occur? Osmosis always occurs from higher water potential to lower water potential. Number D. Which solution has a higher solute potential? Please think it out and drop down in the comment section below. If you failed or face any problem in solving these questions, please let us know and we'll solve them out pretty soon. E. At equilibrium, which chamber will have low water potential? F. If one chamber has psi of minus 2000 kilopascal and the other of minus 1000 kilopascal, which is the chamber that has the higher psi? Number G. What will be the direction of movement of water when two solutions with psi W equals to 0.2 megapascal and psi W equals to 0.1 megapascal are separated by selectively permeable membrane? Please drop down the answers down below in the comment section. Let us discuss another experiment where a solution of sucrose in water taken in a funnel is separated from pure water in a beaker by a selectively permeable membrane. You can get this kind of membrane in an egg. Remove the yolk and the albumin through a small hole at one end of the egg and place the shell in dilute solution of hydrochloric acid for a few hours. The eggshell dissolves, leaving the membrane intact. water will move into the funnel, resulting in rise in level of solution in the funnel. This will continue till the equilibrium is reached. In case sucrose does diffuse out through the membrane, will this equilibrium be ever reached? External pressure can be applied from the upper part of the funnel such that no water diffuses into the funnel through the membrane. This pressure required to prevent water from diffusing is in fact the osmotic pressure, and this is the function of the solute concentration. More the solute concentration, greater will be the pressure required to prevent water from diffusing in. Numerically, osmotic pressure is equivalent to the osmotic potential, but the sign is opposite. Osmotic pressure is the positive pressure applied while osmotic potential is negative. 11.2.3 Plasmolysis. The behavior of plant cells or tissues with regard to water movement depends upon the surrounding solution. If the external solution balances the osmotic pressure of the cytoplasm, it is said to be isotonic. If the external solution is more dilute than cytoplasm, it is hypotonic, and if the external solution is more concentrated, it is hypertonic. Cells swell in hypotonic solutions and shrink in hypertonic ones. Plasmolysis occurs when water moves out of the cell and the cell membrane of a plant cell shrinks away from its cell wall. This occurs when the cell or tissue is placed in solution that is hypertonic to the protoplasm. Water moves out. It is first lost from the cytoplasm and then from the vacuole. Water, when drawn out of the cell through diffusion into extracellular fluid, causes the protoplast to shrink away from the walls. The cell is said to be plasmolyzed. The movement of water occurred across the membrane, moving from an area of higher water potential to an area of low water potential outside the cell. What occupies the space between the cell wall and the shrunken protoplasm in the plasmolyzed cell? Well, it is the liquid medium. When the cell or tissue is placed in isotonic solution, there is no net flow of water towards inside or outside. If the external solution balances the osmotic pressure of the cytoplasm, it is said to be isotonic. When water flows into the cell and out of the cell and are in equilibrium, the cells are said to be flaccid. The process of plasmolysis is usually reversible. When the cells are placed in hypotonic solution or dilute solution as compared to the cytoplasm, water diffuses into the cell, causing the cytoplasm to build up a pressure against the wall that is called turgor pressure. The pressure exerted by the protoplast due to entry of water against the rigid wall is called pressure potential psi p. Because of the rigidity of the cell wall, the cell does not rupture. This turgor pressure is ultimately responsible for enlargement and extension growth of cells. What would be the psi p of a flaccid cell? Which organism other than plant possess a cell wall? 11.2.4 Imbibition. Imbibition is a special type of diffusion when water is absorbed by solid colloids, causing them to increase in volume. The classical examples of imbibition are absorption of water by seeds and dry wood. The pressure that is produced by the swelling of wood has been used by prehistoric men to split rocks and boulders. If it were not for the pressure due to imbibition, seedlings would not have been able to emerge out of the soil into open. They probably would not have been able to establish. Imbibition is also diffusion since water movement is along a concentration gradient. The seed and other such materials have almost no water, hence they absorb water easily. Water potential gradient between the absorbent and the liquid imbibed is essential for imbibition. In addition, for any substance to imbibe in a liquid, affinity between the adsorbent and the liquid is also a prerequisite. 11.3 Long distance transport of water. At some earlier stage, you might have carried out an experiment where you had placed a twig bearing white flowers in colored water and had watched it turn color. On examining the cut end of the twig after a few hours, you had noticed that the region through which the colored water moved, that experiment very easily demonstrates that the path of water movement is through the vascular bundles, more specifically the xylem. Now we have to go further and try and understand the mechanism of movement of water and other substances up a plant. Long distance transport of substances within a cell cannot be diffusion alone. Diffusion is a slow process. It can account for only short distance movement of molecules. For example, the movement of a molecule across a typical plant cell, about 50 micrometers, takes approximately 2.5 seconds. At this rate, can you calculate how many years would it take for movement of molecules over a distance of 1 meter within a plant by diffusion alone? Well, that would be many years. In a large and complex organism, both substances have to be moved long distances. Sometimes the site of production or absorption of site of storage are too far from each other. Diffusion or active transport would not suffice. Special long-distance transport systems are necessary so as to move substances across long distances as much as a faster rate. Water and minerals and food are generally moved by a mass or bulk flow system. Mass flow is the movement of substances in bulk or en masse from one point to the other as a result of pressure differences between the 2 points. It is a characteristic of mass flow that substances, whether in solution or in suspension, are swept along the same pace as in a flowing river. This is unlike diffusion, where different substances can be achieved either through a positive hydrostatic pressure gradient, example garden hose, or a negative hydrostatic pressure gradient, example suction through a straw. The bulk movement of substances through conducting or vascular tissues is called translocation. Do you remember studying cross-sections of roots, stems, and leaves of higher plants and studying vascular system? The higher plants have a highly specialized vascular system like xylem and phloem. Xylem is associated with translocation of mainly water, mineral salts, some organic nitrogen, and hormones from roots to aerial parts of the plant. The phloem translocates a variety of organic and inorganic solutes, mainly from leaves to other parts of the plant. 11.3.1 How do plants absorb water? We know that the roots absorb most of the water that goes into plants. Obviously, that is why we apply water to the soil and not in the leaves. The responsibility of absorption of water and mineral is more specifically the function of root hairs that are present in myelin at the tips of the roots. Root hairs are thin-walled, slender extensions of root epidermal cells that greatly increase the surface area of absorption. Water is absorbed along with mineral solutes by root hairs, purely by diffusion. Once water is absorbed by the root hairs, it can move deeper into the root layers by two distinct pathways: apoplast pathway and symplast pathway. The apoplast is the system of adjacent cell walls that is continuous throughout the plant except at the Casparian strips of the endodermis in roots. The apoplastic movement of water occurs exclusively through intercellular spaces and the wall of the cells. Movement through apoplast does not involve crossing the cell. This movement is dependent on the gradient. The apoplast does not provide any barrier to water movement, and water movement is through mass flow. As water evaporates into intercellular spaces or the atmosphere, tension develops in the continuous stream of water in apoplast. Hence, mass flow of water occurs due to adhesive and cohesive properties of water. The symplast system is the system of interconnected protoplast. Neighboring cells are connected through cytoplasmic strands that extend through the plasmodesmata. During symplast movement, the water travels through the cells, their cytoplasm. Intercellular movement is through plasmodesmata. Water has to enter the cells through the cell membrane, hence movement is relatively slower. Movement is again down a potential gradient. Symplastic movement may be aided by cytoplasmic streaming. You may have observed cytoplasmic streaming in cells of hydrilla leaf. The movement of chloroplast due to streaming is easily visible. Most of the water flow in the roots occur via apoplast, since the cortical cells are very loosely packed and hence offer no resistance to water movement. However, the inner boundary of the cortex, the endodermis, is impervious to water because of the band of suberized matrix called the Casparian strip. Water molecules are unable to penetrate the layer, so they are directed to wall regions that are not suberized into the cell through the membranes. The water then moves through the symplast and again crosses the membrane to reach the cells of the xylem. The movement of water through the root layers is ultimately symplastic in the endodermis. This is the only way water and other solutes can enter the vascular cylinder. Once inside the xylem, water is again free to move between cells as well as through them. In young roots, water enters directly into the xylem vessels and tracheids. These are non-living conduits and so parts of apoplast. The path of water and mineral ions into the root vascular system is summarized in figure 11.7. Some plants have additional structures associated with them that help in water and mineral absorption. The mycorrhiza is a symbiotic association of a fungus with the root system. The fungal filaments form a network around this young root, or they penetrate the root cells. The hyphae have a very large surface area that absorb mineral ions and water from the soil, from a much larger volume of soil that perhaps a root cannot do. The fungus provides minerals and water to the roots. In turn, the roots provide sugar and nitrogen-containing compounds to the mycorrhiza. Some plants have an obligate association with mycorrhiza. For example, pinus seeds cannot germinate and establish without the presence of mycorrhiza. 11.3.2 Water movement up a plant. We looked at how plants absorb water from the soil and move into the vascular tissues. We now have to try to understand how water is transported into various parts of the plant. Is the water movement active or is still passive? Since the water has to be moved up a stem against the gravity, what provides energy for this? 11.3.2.1 Root pressure. As various ions from the soil are actively transported into vascular tissues of the roots, water follows its potential gradient and increases the pressure inside the xylem. The positive pressure is called root pressure and can be responsible for pushing up water to small heights in a stem. How can we see that root pressure exists? Choose a small soft stem plant on a day, and when there is plenty of atmospheric moisture, cut the stem horizontally near the base with a sharp blade. In the morning, you will soon see drops of solution ooze out of the cut stem. This comes out due to the positive root pressure. If you fix a rubber tube to the cut stem as a sleeve, you can actually collect and measure the rate of exudation and also determine the composition of the exudates. Effects of root pressure is also observable at night and early morning when evaporation is low and excess water collects on the tip of the droplets around small special openings of veins near the tip of grass blades and leaves many herbaceous parts. Such water loss in liquid phase is known as guttation. Root pressure can at best only provide a modest push in overall process of transport. They obviously do not play a major role in water movement up tall trees. The greatest contribution of root pressure is to re-establish the continuous chains of water molecules in the xylem, which often break under enormous tensions created by transpiration. Root pressure does not account for the majority of water transport. Most plants meet their need by transpiratory pull. 11.3.2.2 Transpiration Pull. Despite the absence of heart or a circulatory system in plants, the upward flow of water through the xylem in plants can achieve fairly high rates up to 15 meters per hour. How is this movement accomplished? A long-standing question is whether water is pushed or pulled through a plant. Most researchers agree that water is mainly pulled through the plant and that the driving force for this process is transpiration from the leaves. This is referred to as cohesion tension transpiration pull model of water transport. But what generates this transpiration pull? Water in transit in plants. Less than 1% of the water reaching the leaves is used in photosynthesis and plant growth. Most of it is lost through the stomata in the leaves. This water loss is known as transpiration. You have studied transpiration in an earlier class by enclosing a healthy plant in polythene bag and observing the droplets of water formed inside the bag. You could also study water loss from a leaf using cobalt chloride paper, which turns color on absorbing water. 11.4 Transpiration. Transpiration is the evaporative loss of water by plants. It occurs mainly through stomata. Besides the loss of water vapor in transpiration, exchange of oxygen and carbon dioxide in the leaf also occurs through these stomata. Normally, stomata are open in the daytime and closed during the night. The immediate cause of opening and closing of stomata is the change in turgidity of the guard cells. The inner wall of each guard cell towards the pore or stomatal aperture is thick and elastic. The turgidity increases within the two guard cells flanking each stomatal aperture or pore. The outer thin walls bulge out and force the inner walls into a crescent shape. The opening of stomata is also aided due to the orientation of microfibrils in the cell walls of the guard cells. Cellulose microfibrils are oriented radially rather than longitudinally, making it easier for the stomata to open. When the guard cell lose turgor, Due to water loss, the elastic inner walls regain their original shape. The guard cells become flaccid and stoma closes. Usually, the lower surface of a dorsiventral leaf, often dicotyledonous leaf, has higher number of stomata, while in an isobilateral or monocotyledonous leaf, they are about equal on both the surfaces. Transpiration is affected by several external factors: temperature, light, humidity, wind speed. Plant factors that affect transpiration include number and distribution of stomata, percent of open stomata, water status of the plant, canopy, structure, etc. The transpiration-driven ascent of xylem sap depends mainly on the following physical properties of water. Number 1. Cohesion, that is mutual attraction between the water molecules. Number 2. Adhesion, that is attraction of water molecules to polar surfaces such as the surface of the tracheary elements. Number 3. Surface tension. Water molecules are attracted to each other in the liquid phase more than in water in the gaseous phase. These properties give water high tensile strength, that is an ability to resist the pulling force, and high capillarity, that is the ability to rise within the tubes. In plants, capillarity is aided by small diameter of the tracheary elements, the trachees and vessel elements. The process of photosynthesis requires water. The system of xylem vessel from root to the leaf vein can supply the needed water. But what force does a plant use to move water molecules into the leaf parenchyma cells where they are needed? As water evaporates through the stomata, since the thin film of the water over the cell is continuous, it results in pulling of water, molecule by molecule, into leaf from the xylem. Also, because of lower concentration of water vapor in the atmosphere as compared to the substomatal cavity and intercellular spaces, water diffuses into the surrounding air. This creates a pull. Measurements reveal that the forces generated by transpiration can create pressure sufficient to lift a xylem-sized column of water over 130 meters high. 11.4.1 Transpiration and photosynthesis, a compromise. Transpiration has more than one purpose. It creates transpiration pull for absorption and transport of plants, supplies water for photosynthesis, transports mineral from the soil to all parts of the plant, cools leaf surfaces sometimes 10 to 15 degrees by evaporative cooling, maintains the shape and structure of the plants by keeping the cells turgid. An actively photosynthesizing plant is an unsatiable need for water. Photosynthesis is limited by the available water that can be swiftly depleted by transpiration. The humidity of rainforest is largely due to its fast cycling of water from root to leaf to atmosphere and back to the soil. The evolution of C4 photosynthetic system is probably one of the strategies for maximizing the availability of CO2 while minimizing water loss. C4 plants are twice as efficient as C3 plants in terms of fixing carbon dioxide. However, a C4 plant loses only half as much as C3 plant for the same amount of CO2 fixed. 11.5 Uptake and transport of mineral nutrients. Plants obtain their carbon and most of the oxygen from CO2 in the atmosphere. However, the remaining nutritional requirements are obtained from water and minerals in the soil. 11.5.1 Uptake of mineral ions. Unlike water, all minerals cannot be passively absorbed by roots. Two factors account for this. Number 1, minerals are present in soil as charged particles which cannot move across cell membranes. And number 2, the concentration of minerals in the soil is usually lower than the concentration of mineral in the root. Therefore, most minerals must enter the root by active absorption into the cytoplasm of the epidermal cells. This needs energy in the form of ATP. The active uptake of ions is partly responsible for the water potential gradient in roots and therefore for the uptake of water by osmosis. Some ions also move into the epidermal cells passively. Ions are absorbed from soil, both active and passive transport. Specific proteins in the membranes of root hair cells actively pump ions from the soil into the cytoplasm of the epidermal cells. Like all cells, the endodermal cells have many transport proteins embedded in the plasma membrane. They let some solutes cross the membrane but not others. Transport proteins of endodermal cells are control points where a plant adjusts the quantity and type of solutes that reach the xylem. Note that the root endodermis because of the layer suberin has the ability to actively transport ions only in one direction. 11.5.2 Translocation of mineral ions. After the ions have reached xylem through active or passive uptake or a combination of the two, that further transport up the stem to all plants of the plant is through transpiration stream. The chief sinks for the mineral elements are the growing regions of the plant, such as the apical and lateral meristems, young leaves, developing flowers, fruits and seeds, and the storage organs. Unloading of mineral ion occurs at the fine vein endings through diffusion and active uptake by these cells. Mineral ions are frequently remobilized, particularly from older senescing parts. Older dying leaves export much of the mineral content to younger leaves. Similarly, before leaf fall in deciduous plants, minerals are removed to other parts. Elements most readily mobilized are sulfur, phosphorus, nitrogen, and potassium. Some elements that are structural components like calcium are not remobilized. An analysis of the xylem exudates show that though some of the nitrogen travels as inorganic ions, much of it is carried in the organic form as amino acid and related compounds. Similarly, small amounts of phosphorus and sulfur are carried as organic compounds. In addition, small amount of exchange of materials does take place between xylem and phloem. Hence, it is not that we can clearly make a distinction and say categorically that xylem transports only inorganic nutrients while phloem transports only organic nutrients, as was traditionally believed. 11.6 Phloem transport flow from source to sink. Food, primarily sucrose, is transported by the vascular tissue phloem from source to sink. Usually, the source is understood to be the part of the plant which synthesizes the food, that is the leaf, and the sink the part that needs to store the food. But the source and sink may be reversed depending on the season or the plant's need. Sugar stored in root may be mobilized to become a source for food in early spring in roots when the buds of trees act as sink. They need energy for growth and development of photosynthetic apparatus. Since the source-sink relationship is variable, the direction of movement in phloem can be upwards or downwards, that is, bidirectional. This contrasts with that of xylem where the movement is always unidirectional, that is, upwards. Hence, unlike one-way flow of water in transpiration, food in phloem sap can be transported in any required direction so long as there is a source of water and a sink able to use, store or remove the sugar. Phloem sap is mainly water and sucrose, but other sugars, hormones and amino acid are also transported or translocated through the phloem. 11.6.1 The pressure flow or mass flow hypothesis. The accepted mechanism used for translocation of sugars from source to sink is called pressure flow hypothesis. As glucose is prepared at the source, by photosynthesis it is converted into sucrose, a disaccharide. The sugar is then moved in the form of sucrose into companion cells and then into living phloem sieve tube cells by active transport. This process of loading at the source produces a hypertonic condition in the phloem. Water in the adjacent xylem moves into the phloem by osmosis. As osmotic pressure builds up in the phloem, the sap will move to areas of lower pressure. At the sink, osmotic pressure must be reduced. Again, active transport is necessary to move the sucrose out of the phloem sap into the cells which will use the sugar, converting it into energy, starch or cellulose. As sugars are removed, the osmotic pressure decreases and water moves out of the phloem. To summarize, the movement of sugars in the phloem begins at the source, where sugars are loaded, actively transported into the sieve tube. Loading of phloem sets up a water potential gradient that facilitates the movement in the phloem. Phloem tissue is composed of sieve tube cells, which forms long columns with holes in their end walls called sieve plates. Cytoplasmic strands pass through the hole in the sieve plate, so forming continuous filaments. As hydrostatic pressure in the sieve tube of phloem increases, pressure flow begins. The sap moves through the phloem. Meanwhile, at the sink, incoming sugars are actively transported out of the phloem and removed. As complex carbohydrates, the loss of solute produces a high water potential in the phloem and water passes out, returning eventually to xylem. A simple experiment called girdling was used to identify the tissues through which food is transported. On the trunk of a tree, a ring of bark up to a depth of phloem layer can be removed carefully. In the absence of downward movement of food, the portion of bark above the ring of the stem become swollen after a few weeks. This experiment shows that the phloem is the tissue responsible for translocation of food and that transport takes place only in one direction, that is towards the roots. This experiment can be performed by you easily. Summary. Plants obtain a variety of inorganic elements and salts from the surroundings, especially from water and soil. The movement of these nutrients from environment into the plant as well as from one plant cell to the other plant cell essentially involves movement across the cell membrane. Transport across cell membrane can be through diffusion, facilitated transport or active transport. Water and minerals absorbed by roots are transported by xylem and organic material synthesized by leaves is transported to other parts of the plant through phloem. Passive transport and active transport are two modes of nutrient transport across cell membranes in living organisms. In passive transports, nutrients move across the cell membrane by diffusion, without any use of energy, as it is always down the concentration gradient and hence entropy driven. This diffusion of substances depends on the size, solubility in water or organic solvents. Osmosis is a special type of diffusion of water across a selectively permeable membrane which depends on pressure gradient and concentration gradient. In active transport, energy in the form of ATP is utilized to pump molecules against a concentration gradient across membranes. Water potential is the potential energy of water molecule which helps in movement of water. It is determined by the solute potential and pressure potential. Osmotic behavior of cell depends on the surrounding solution. If the surrounding solution of the cell is hypertonic, it gets plasmolysed. The absorption of water by seeds and dry wood takes place by a special type of diffusion called imbibition. In higher plants, there is a vascular system comprising of xylem and phloem responsible for translocation. Water, minerals, and food cannot be moved within the body of a plant by diffusion alone. They are therefore transported by a mass flow system. Movement of substances in bulk from one point to the other as a result of pressure differences between the 2 points. Water absorbed by root hairs moves into the root tissue by two distinct pathways, that is apoplast and symplast. Various ions and water from soil can be transported up to a small height in stem by root pressure. Transpiration pull model is the most acceptable to explain the transport of water. Transpiration is the loss of water in the form of water vapors and from the plant parts through stomata. Temperature, light, humidity, wind speed, and number of stomata affect the rate of transpiration. Excess water is also removed through tips of leaves of plant by guttation. Phloem is responsible for transport of food, primarily sucrose, from the source to the sink. The translocation in phloem is bidirectional. The source-sink relationship is variable. The translocation of phloem is explained by the pressure flow hypothesis. So that's it for today. See you again in the next one. Till then, subscribe to the channel and stay tuned. Thank you.